Okay, good morning, students. Few students are still to join, but I think we can start with the class. So today we are going to start a new topic and a very important topic actually, lab safety. So lab safety is important from the point of view that we all, as a science student, we work in a lab in one form or the another form, and there are many chances that sometimes accidents they take place due to the ignorance of the students. Sometimes probably when you are new to the lab, you don't know many chemicals, you don't know, I mean, whether to touch few things or not, whether to smell few chemicals or not. There are many things there in the lab, and many unlabeled solutions also sometimes. So it is very dangerous, right? So as we enter the lab, so first thing which we tell students usually is the lab safety and the lab precautions, especially in a microbiological labs or a molecular biology labs or a labs where a lot of work on the pathogenic organisms is also being done sometime, right? So how we should behave in the lab, what should be the journal guidelines in the lab. So since you all have been working in the lab, one or the other form, maybe normal lab, so I'm sure you all must be aware of the good microbiological practices, right? So I would like to hear it from you that what do you think, I mean, how you should behave in the lab, what kind of a behavior should be expected from a researcher or a student when they enter any kind of a research lab or a teaching lab. Yes, who's going to tell me this? Yes, Manaswani, switch on your camera and tell me.
Ma'am, so am I visible? Yeah.
Yeah, you're visible. Yes.
So basically, when a researcher is entering a lab, the first thing
Mm-hmm.
is that they should have, the equipment which protects themselves. So let's say like safety goggles and lab
Mm-hmm.
coats and gloves and all. And another important point is that they should have proper knowledge of the MSDS, which is the material safety data sheet. So they should know, exactly what chemicals and equipment that they're working with, and what way they should be handled, and in case of any kind of accident, what the side effects are and how they can heal or cure those side effects or handle it. But yeah, basic information of the workings of the lab is very important.
Yes, yes, Manaswini, you're right that as soon as when we enter the lab, we should know about the kind of chemicals we are going to work with. Yes, Shakti, please, your views, switch on your camera.
Yes, ma'am. Am I visible?
Yes, you are visible.
Oh, yes, ma'am. So, personal protection is very important. I think once Devendra sir asked me like why you wear shoes in like laboratories. So to protect ourselves from viruses or the chemicals they
Mm-hmm.
may fall on your legs if you're not wearing proper
Yes,
shoes. And
yes.
also, ma'am, like we should always look, as Manaswini said, like what chemicals we are using. We should know about that. What we cannot mix, like we cannot mix inorganic chemicals with organic chemicals that can lead to
Mm-hmm.
explosions. So
Yeah.
that knowledge is very important. And also, if something happens, then how you can control that. Like if you got got fire in your hand, then you should not
Mm-hmm.
run away. Like that will provide air.
Mm.
You should
Right.
rub it on the floor. So I think that all should be known, and that is important.
Yeah, yeah, that's right, Shakti and Manaswini. You both are right that basic when you enter the lab, if you don't know something, ask your supervisor, ask the person who is demonstrating the experiment or who is taking your experiment, instead of experimenting with different chemicals yourself. So you should be very careful when you are doing. There are many things. There are many equipments in the lab, such as autoclave. We don't leave autoclaves unattended. We need to check the water level continuously. We need to see the pressure gauge is rising properly. Sometimes, these things are automatic, but sometimes it happens that they are not working properly, and due to which the pressure can keep on increasing. So at the end, these things can lead to the blast of the autoclave also. So if person is sitting nearby and carefully observing the autoclave, so he or she can switch off the autoclave from the backside, so that, I mean, the pressure is not rising beyond a certain limit. Usually there are auto cuts available, but sometimes accidents do happen, right? So also, like Shakti said, that there are material safety data sheets, and Manasni also said that material safety data sheets which are available in the lab for all the chemicals. So before you start working on a particular chemicals, at least read about it, right? Not all chemicals which are present that in the lab that we use are hazardous, but yes, some are very corrosive, some can cause our health hazards also, some can cause the physical hazards also. So we should know about these chemicals. And the third important point is that okay, now you are careful enough, but sometimes, yes, accidents do happen, right? How much careful we are, but sometimes it happens. Then you should know that whom to report, what to be done next, where is the safety first aid kits are, where they are located, whom to contact, and how to deal with those kind of accidents. So it is very important. So smaller precautions taken while we are working in the lab can prevent a larger or any small accident also. Like there's a very small common thing, and I've been always telling my students this when we start with the microbiological experiment, that when you work in a laminar flow, it is very, we always in the laminar flow, when we are dealing with the microorganisms or culturing or a plating or anything like that, so we are very careful about the contamination, because we know that if our hands are not clean, we can contamination can be passed on to the plates, and results will not be proper. So it is usually recommended that before you pour your plate or your streak, you just wipe off your hand with the spirits, right? So sometimes it happens that students, they use a large amount of a spirit, or by chance also the more than required amount of the spirit fell in your hand, and you wipe off, you just rub your hands, and immediately you just try to light up the Bunsen burner or something like there for the again the safety purposes, so that contamination is not. But in such cases, and this has happened also, that sometimes the hands, they caught fire, because the spirit is still in your hand and it is very flammable, right? So it can caught fire. So these are the small things that we need to take care, that we don't light up the burner before our hands are really very dry and we don't have much spirit left on our hand, so that it can catch fire. We should be careful when we are dealing with the molecular biology. Certain molecular biology agents are, as I told you in the last lecture also, they have certain cancerous properties, also carcinogenic, so we should be wearing gloves. So basically lab safety, or this lecture or topic that we are going to cover in 2 or 3 lectures, it deals with how you behave in the lab, journal lab behavior, or a good microbiological techniques or practices, or how we deal with the pathogenic microorganisms. So pathogenic microorganisms is definitely being done in the research teaching lab, but they are being done in the research lab if we are working on different kind of a pathogen. So how what kind of a biosafety levels are recommended, what kind of a containment facilities should be there, what kind of a lab should be there, and how the risk groups like pathogens, they are being categorized in different risk groups according to the diseases they are causing, according to the threat, whether they are causing individual threat or a community threat, and how much to how much level, whether any therapeutic agent is available, any therapeutic method is available, or vaccination is available if such threat happens, or some, or when, how we deal if we manipulating some new pathogen, or we are handling some new pathogen which we don't know much, so how careful we should be for that. So this is the lecture which deals with the journal lab practices, how we should be careful, what how we can take different precautions, and what are different biosafety levels, and what are different risk group pathogens which are directly associated with the biosafety level. So we'll try to cover this in 2 or 3 lectures. Let's try how we finish this off. So very journal lecture, and most of the thing I think you all know, because you all have been studying microbiology 1 and 2, and there also a lot of guidelines are being provided that how to be very careful while you are working in the lab. So I think most of the things should be the repetition of the which you already know, right? So working in the lab is definitely good experience whenever, especially for the graduates, whenever after school, because in the school we don't do much of the practicals, more of the theory is there. Some practicals in 10 plus 1 and plus 2, and especially for the science students, the practicals are a very important part. So as soon as we get into the graduation, the exposure to the lab increases, right? So yes, we know how we can do things with our own hands. We know how to prepare media, how to do plating, how to extract RNA or DNA. So definitely it is very exciting process also, that we are more enthusiastic to learn new things, something new. We can isolate a DNA. So this is very fascinating, that how we can isolate a DNA and then how we can actually see it, whether we have isolated it correctly or not. So definitely it is a very good experience. But then lab working in a lab can cause threats and hazards also, which a classroom teaching does not have. So classroom teaching, in a classroom we don't face such kind of a hazard. It's a safe environment where we go, we sit, lecture is being given, so there's nothing sort of like hazardous chemical, or you don't deal with the pathogens or any corrosive chemical or any reagent, right? So it is very important to know your surroundings. We should know where is the entry of the room, where is the exit of the room, if there is any emergency, if there are more than one exit. So we should know about that also. And also other things that we need to know are about where are the fire extinguishers kept. So usually every year they do a fire drill. So if this is a fire department in Amity also, so they do a mock drill basically. So there they create a such environment that there's a fire alarm being, then how, which way we should follow to exit. And they also tell everyone how to use fire extinguishers in case of an emergency if required. So these drills, they play a very important role in educating students, teachers, and staff in the university also. So in order to, your proper training is very important. And the first important thing is to evacuate the area, that in a proper way, how we should do it. Then where are the fire alarms, which are, if they are kept in the laboratory or outside the laboratory, we should know their position. So labs, they have a hazardous substance. So their hazardous substance is basically any material or a substance that can cause you a health hazard and a physical hazard, both, right? Chemicals and biological agents. So hazardous substances include any chemicals also, and biological agents, which are your pathogens, which can cause diseases. And then another term is there, biohazard. So biohazard, as the name bio indicate, that any organism which can cause replication and capable of causing disease in human, animal, and plant. So biohazard is dealing with the pathogen, biological. A bio term is applied before the hazard. So it include only the pathogens which can cause diseases in human, plants, and animal. So what kind of a health hazard could be there? They could be, the chemical could be a carcinogen, it could be toxic, it could be irritant, irritation to your skin, it could be corrosive also, could be hepatotoxin, depending upon that, like if it is affecting your liver, hepatic toxin, if it could be a nephrotoxin, if it is affecting your kidneys, it could be a neurotoxins, right? So there are different kind of chemicals which have a different kind of properties, and they may affect your different organs differently. Then physical hazard could be explosives, such as your flammable substances mostly, which we need to be very careful while using them. They could be some gases, compressed gases, some unstable materials, or water reactive materials. So we should be very careful while using such chemicals or a material. Or this, for example, there's a liquid nitrogen. So in liquid nitrogen, we use very commonly in the molecular biology labs while extracting DNA or RNA, right? So it frees the substance at sometimes to store cell lines also, or store some cultures also. We use a liquid nitrogen. So it has a property that it will freeze the things immediately, right? Without letting it thaw or any other form, it will just immediately freeze that. So when we do crushing of the sample, it is easy to make them into powder form. But then liquid nitrogen can give you burns, like heat burns that we have, so cold burns. So it can burn your skin if it we come directly in contact with the liquid nitrogen. So there are special gloves, which are very thick gloves that we need to use while we use the liquid nitrogen. We need to use some goggles also, because the splashes can go to our eyes also. So we should be very careful while we are using liquid nitrogen also in the lab. So while there are a number of physical hazards and health hazards are there, so sometimes our eyes are the most sensitive part in our face. So if something goes to our eyes, some kind of a substance or a chemical, so we should know if there are any eye wash which is located. Usually these are located in the high-end labs, because where they are being required, or we are dealing with such kind of a chemicals, or just a sink where clean water is there and where we can quickly go and wash off our eyes very fast. Then first aid kits. So like Kinevity also, we have a first aid kits in the labs. Every lab they have a first aid kits, or they are kept with the lab uh, coordinator or a faculty. So we should know that in this, if you're working in one lab, so where is the lab kit kept, and where they are kept in, with which faculty is having that kit, or if in some common area they are being placed, then we should know the position. And then for after the first aid kit, yes, clinics are in every institution, so we should know how to. We should first inform the supervisor, and then if required, we can go to the clinic also. Some kind of a medical intervention is required. So hazards are biological, chemical, or a radioactive material which can cause a health hazard. So radioactive material is another kind of a hazard which is not very commonly used in the teaching labs. We don't recommend that. It is being always used in the research labs, and there are also separate rooms to use such kind of a material, and a lot of precautions are being required when you radioactive material. Basically, when we do this northern or southern blotting, so we label the probe with the radioactive material for radioactive detection finally, right? So the results in those cases, the signals that we obtained at the end are very clear. But then this process is little dangerous. So we use another process. We use enzyme labeled kit these days. So those kits are safer to be used. Yes, the results are not as good as we use when we do the radioactive labeling, but still, safety wise, it is being preferred. Then biological hazards, as I told you, any organism or a material or a biological origin. Then infectious agent, definitely any organism which can cause infection or a disease in human, animal, or a plant. Then we also deal with the recombinant DNA. This is a very common experiment these days. We do these kind of experiment. So we all know that RDA or recombinant DNA is any molecule that are being being constructed outside living cell. We artificially create them, we synthesize them, we synthesize any particular gene, and then we introduce into the some other organisms, right? So such kind of manipulations are also not done in open, and we need to be careful while we are doing such kind of a work. Then infectious agents are definitely harmful, because they can cause illness, and in extreme cases death also. So we need to be very careful while we are handling them. So there should be, dealing with the infectious pathogen, a separate training is being required. In a teaching lab, we don't handle such pathogens, so we don't go for such things. But when we do research in the higher level for your PhD maybe, or at the for some there are projects, some research is going on, there are special instructions, and the people who work in such areas, they are being informed how to deal with those pathogens. So depending upon the organism we are working, will what kind of a facility that will use, that is called a biosafety levels. So if we are working with the risk group 1 pathogen, we can deal it with biosafety level 1. And if we are working with risk group 4 pathogen, we'll deal it with in the biosafety level 4. So there's another term called containment. So containment is basically confinement of a biohazardous agent that is being either cultured or manipulated or stored or transported in order to prevent or limit its contact with the people or the environment, right? So containment, right? We are putting it in a separate place in a proper closed area where the access to the normal people is not there. So when we deal with the pathogenic organisms, we usually use such kind of a containment facility also. So during COVID-19, the word containment was very often used. So who is going to tell me what does that containment meant during COVID-19? There were many containment zones which were identified. What does that mean? Why was that done?
Ma'am, it was those zones with
No, Manaswani, stop,
okay,
Manaswani.
okay.
Please, other people, every time Manaswani is answering, I'm not teaching Manaswani only, I'm teaching all of you, right? And we all have gone through this COVID-19 equally. So I think ये जो न्यूज में इतना commonly containment word होता था, that society is declared as a containment society. तो what does that mean? Yes, देवांशी, क्या मतलब था इस बात का? देवांशी, I'm asking you. Ishita? No, this is not right, students. You don't answer, right? अब मैं सबको ना कैमरा ऑन करवा के and then I'll take class so that I can see what exactly you all are doing and I can ask anyone. Yes, Athoiba, what does containment mean?
Ma'am, am I audible?
Yes, containment.
Like, containment, action that prevents the hello.
Hello, yes, Athoiba, I can't hear you.
Ma'am, like,
Yes. Okay, fine. Yes, Kshitij, what does containment means? What does it meant during COVID-19? Why we were saying that?
Keeping something harmful substance
Separated. So why we were doing containment zones क्यों बना रहे थे हम जब COVID-19 था? क्या फायदा हुआ उससे? क्या फायदा था उससे? What was its use? Yes, Sahiba. I'm going to mark you all absent for today's class if you don't answer.
Yes, ma'am. Good morning, ma'am.
हाँ साहिबा, कंटेनमेंट जोन्स का क्या मतलब था जब हम कोविड-19 में कंटेनमेंट जोन्स बना रहे थे? Devanshi, I don't need your written answer. I wanted you to switch on your camera and answer directly. Yes, Sahiba.
Ma'am, I'm sorry, ma'am. I can't recall at this time.
क्यों साहिबा, it was so easy. कंटेनमेंट जोन्स बन गए थे दिल्ली में, so many societies were being contained. क्या मतलब था उसका? क्या गाइडलाइंस थे गवर्नमेंट के उस कंटेनमेंट जोन के लिए? बताओ मुझे जरा. Use your brain. This is so simple. हाँ.
Ma'am, it was like कि जहाँ मतलब जो कंटेनमेंट जोन बन गए थे तो वो लोग को मना करा गया था that others should not go there and they should not come out of that zone.
Yes, Sahiba. So this is a simple answer, right? People were, and why this was done? Yes, Tridev, why this was done?
This was done to limit the virus. Basically, it was expanding. That's why it was done, so that
yes, the transmission rate of the virus was so much that people who were coming in contact with the infected people who were coming in contact with the healthy one, they were also getting infected, right? So they wanted to break the chain of the transmission. So what did the government do? They just, if in particular society more than 5-10 people are found infected with a particular virus, they contain the society. Containment का मतलब है they just stopped. Nobody will go inside, nobody will come outside. A requirement or grocery will be provided at the doorstep so that virus is contained in a particular area, right? Till that incubation time is being over of that virus, so that it is not multiplying further, or those people were found negative by some kind of a report. So they were containing the virus, they were limiting the spread of the virus. So that means containment. So here also the containment means we are limiting the access of that particular pathogen, which is infectious, to a one particular place. And that place should not be accessible to the people outside the laboratory, people who are not working in that area. We are containment, we are, so that it does not spread out, right? So methods which are used for containment are physical and biological barriers. There are different physical barriers and different biological barriers that can be used, right? So yes, chemicals can pose a lot of hazards. So chemicals which generate harmful fumes are required to be used in a proper fume hood. There are fume hoods which especially are designed in such a way, you open the bottle in a fume hood, you'll not put your face inside that. So if any fumes are being generated, they are directed to outside through a exhaust fan. There's a pipe, a big pipe which just takes directly it to the outside, so we don't get in direct contact with the chemicals. So chemicals can release these kind of fumes which can not only harm the environment but also kind of major health threats. So they are being sometimes the filters are being provided so that they are being filtered before they are being released outside. So we should fume hoods are basically for the chemicals such as HCLs, which generate a lot of fumes, and they should be opened properly, right? Then the next point is a lab attire. What does that mean? That means what kind of your clothing should be in the lab, what should be you wearing. So you should no open toed shoes covering, you should be wearing shoes which cover your full feet, no short dresses. If yes you're wearing, then full lab coat should be there. And the ethics of working in a lab is you tie up your hairs properly if you're working, or you wear a head covers. And what personal protective equipment is important, that the lab coat is the thumb rule. Whether you work any kind of a plant lab, animal lab, or a microbiological lab coat is a thumb rule, that you always wear a lab coat when you are in the lab, so that if any spill happens or anything, so it does not go directly to your body. The lab coat is there to protect. Even the stains, you do a lot of staining experiments, you make a lot of slides during your BSc practicals also. So sometimes there also those stains will then spoil your clothes. So these lab coats, they are a kind of a gap between your body and directly with the chemicals that you are using. It will protect you. We don't eat, smoke, or drink in the lab. It is not at all allowed. We don't store any kind of eating material in the lab refrigerators where we store the chemicals. We do not mouth pipe it in the lab, because it happens many times that accidentally we can just suck out the liquid or any buffer that we are mouth pipetting, and definitely it will be harmful. Then washing hands before leaving the lab or after handling any kind of a material, whether it's contaminated or non-contaminated, it is important. So safe practices, we do not, again there are certain guidelines, we do not use chipped or a cracked glassware, because if we are using glasswares, if for example we are trying to make some media, so we need a flask or some kind of a glassware. So if they are cracked or a chip, so further heating will again, crack will be increased, and they can be broken also. And if you are working directly with them, they can, cuts can be on our hands also. So it is very important that we should not use them. Then if you're working with any kinds of a pathogenic or a hazardous material, there should be second person nearby us, so that if any accident, anything happens, there should be someone to report that. Then lab should be very neat and clean. Clean your working area after you leave the lab. Things should be at their own places from where you have taken them. Decontaminate if required. And decontaminations mean if you're working on a certain kind of a pathogen. For example, when we work in the microbiological lab, we do certain streaking or a plating to culture the pathogens. So before we discard them, after our work is done, we decontaminate them. So decontamination can be done by the autoclaving also. So for the pathogens or organisms, microbes, we recommend the autoclaving for the decontamination, right? Then labeling. The second important part is the labeling, that if we are preparing any buffer, we should label it properly. All chemicals which we procure from outside, they are definitely being labeled, so no need for them. But then we to do any kind of for example, if we are going for a DNA extraction only, so we need to prepare a CTAB buffer, so it should be properly labeled. We do a gel electrophoresis, we need a TBE or TAE buffer. So we should label it properly. And this was the very common mistake while we were also doing experiments in our during our PhD time. The TAE buffer is almost look like a water, just transparent. So if it is not labeled, usually the distilled water bottle and the TAE, they were kept in the similar places, right? So if TAE is not labeled, or we don't label that it's a distilled water, sometimes we can just use mistook TAE for the distilled water and water for the TAE. So after only we run the gel and we don't see anything happening, we realize that we have used water instead of a TAE, because the TAE buffer does not have any kind of a smell and neither any kind of a color. So that's why labeling is very important. We make lot of buffers, we make lot of stock solutions, so it should be properly labeled. So the most dangerous substance is one that has no label on it available. So if we see certain substance in the lab or bottles or a flask which are not labeled, do not touch them or do not use them, just leave them as such. Labeling is also required for chemicals that are in some containers. Sometimes some chemicals are there put in secondary container, so we don't come in direct contact with them. So hazardous chemical, what kind of a hazardous chemical could be there, we need to identify them. Hazard warnings should be available on them, and these things are already available on the containers. Actually, we don't need to do much about them. So another point is the material safety data sheets. So material safety data sheets are available for any kind of a chemical that we procure from outside to work in the lab. So all those chemicals are being, their information is in the material safety data sheets which are present in the lab, whether they are, what kind of a hazard they can cause if we got exposure to them, and what should be the first aid. So all those kind of information about the chemical is being provided in them. So this MSDS will communicate the information necessary regarding hazards which are associated with chemicals and biological agents also. So each component is, again, there is a different guidelines. So it tell us about the kind of a chemical, kind of a hazard it can cause, and then another section which deals with the if we get exposure to these chemicals, how we should deal with this kind of a hazards. Then there is a personal protective equipment. So this PPE term also we have used very quite often during the COVID-19, that we were short of PPE, doctors were not having proper PPE to deal with the patients, right? So there are guidelines to deal with every kind of a pathogen, right? So coronavirus, the transmission rate was very high. So doctors, they need to wear a full coverall gown, which come under protective equipment, and when they are dealing or they are with the patient, so that they themselves do not get contaminated or they do not contract the pathogen from the patient. So PPE is a short form for personal protective equipment. Then all gloves, safety glasses, lab coats, shoe covers, respirators, all these things, they come under the PPE only, personal protective equipment. So knowing what to do, when to use, is very important. That what kind of a PPE is we need to use while we work in a lab, while we deal with the animals for a research purposes, while we deal with the pathogenic organisms. So this is also very important. So chemicals, they are corrosive. So lab sorry, and then gloves, in this case safety glasses, gloves and respirator if required we can use. Then biological agents as well as recombinant DNA can pose the serious health problem. So containment and proper handling techniques are best method to minimize the risks that are associated with these hazards, and PP is the only way that can protect you from the pathogens. So safety glasses, gloves, lab coat, respirator, again depending. Then we sometimes we work with the animals also, like for our research purposes, we need a lot of model animals, so they can scratch and they can bite. Some animals can transmit the zoonotic diseases also. So for them also, we need to use PPE, gloves, lab coat, glasses, respirator, shoe covers, we need to use. Then arthropods are vectors of many important viruses, and sometimes we are working on these arthropod vectors in the lab. So we should be very careful that they do not escape from the closed area, And when we are working with them again, then certain kind of a PPE we should be wearing, which are being recommended, right? So these are just example that personal protective equipments which were recommended for the Ebola virus and the people who were dealing with this virus. So healthcare workers who were caring for a patient with Ebola, they should have a proper comprehensive training is required. Then PPE that covers the clothing, skin, and completely protects the mucous membrane is required when caring for the patients with Ebola. So they should be properly wearing all kind of in this case also the cover all gowns are being required, and respirators if mentioned, people should be wearing that. Then what kind of a PPE for COVID-19? Again, goggles, face shield, mask, glove, cover all gowns, head cover, shoe cover. So people who were dealing, I don't know if any of you have gone for that corona testing, also sample giving. So that person who's taking sample is usually wearing the PPE, and he's covering head to toe, and goggles he's wearing, and from very far away through that long stick he's taking the sample, right? So a lot of carefulness, lot of precautions are required. And face shield and goggles, contamination of mucous membranes of the eyes, nose, and mouth is likely in the scenario of droplets which are generated by cough, sneeze of an infected person. The aerosols are being generated. So it is very important that we cover all these areas. So protection of the mucous membrane of the eyes, nose, mouth by using face shield, goggles is very important when any person who is dealing with the COVID-19 patient. Mask, glove, overall gowns, again, they were recommended. Shoe covers are recommended. Shoe covers should be made of impermeable fabrics. Then head cover should be there, right? So these are again the certain guidelines which were basically for the dealing with the COVID-19 patients in the OPD. So how, what kind of a mask one should wear, what kind of a precautions one should take, right? So today's lecture focused upon the lab safety, general lab safety, different terms such as biohazardous and hazardous material, health hazards and the physical hazards, PPE, MSDS, and guidelines which we take while dealing with the COVID-19 patients or Ebola patients. So that's all for today's lecture. If any clarification is required, please you can ask your question. In the meanwhile, I'm okay. Just a moment. I'm trying to publish the quiz. One small question, basically. So all of you can do that question on which is based on today's lecture only. It is in the form of MS form, so you can just directly answer there. You don't have to upload and scan. Okay, I have assigned it. Maybe within few seconds you'll be able to see that, and just quickly just complete this and submit it right now. Okay. Okay, they are published. All of you, please do it and submit it right now. Just one question, 3-4 lines or 5 lines max. Okay, in the meanwhile, you're submitting your assessment. Please switch on your cameras for attendance also. Yes, Adhish Mishra.
Present, ma'am.
Athoiba.
Ma'am.
Ayushi.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi. Ishita.
Present, ma'am.
Janvi.
Present, ma'am.
Kshitij.
Present, ma'am.
Manaswini.
Present, ma'am.
Namrita.
Present, ma'am.
Namrita क्या Nandini. Nandini.
Present, ma'am.
Nehal.
Present, ma'am.
Rupal.
Present, ma'am.
Sahiba.
Present, ma'am.
Sanjana.
Present, ma'am.
Shakti.
Present, ma'am.
Shivani.
Present, ma'am.
Tanvi.
Present, ma'am.
Tridip. Vivek.
Present, ma'am.
Yukta. Yuvraj.
Present, ma'am.
Devanshi, why your microphone and camera is not working? If there is something wrong, please get it done, right? You need to speak your attendance and you need to switch on your camera, all of you. So make sure it's working fine or properly, right? So please submit your assignments by today also, today itself, that don't leave it up to the tomorrow. And tomorrow also we have a class, we'll again talking about the biosafety, another part where we will be talking about the risk groups and the biosafety levels, right? So thank you so much. We'll meet tomorrow then. Thank you.
Thank you, ma'am.